All right, here we go. We are on day 6, second week of the class. It's actually going to start going by pretty quickly now. Today, I want to cover the blood and blood vessels, and you get to do your blood typing test. I think I had a student who actually was very eager, did the blood typing test this past weekend. I'm curious to know how that turns out. So I'm sure I will find out. And then tomorrow, so this is all part of the cardiovascular system, obviously. The cardiovascular system transports blood, but I thought it was important to talk about what blood is before we actually talk about the cardiovascular system. So today is just blood and blood vessels. Tomorrow we'll talk about the heart and how a heart contracts, which is pretty interesting. Right, so these are all of the functions of blood to deliver oxygen and nutrients, transport waste, transport hormones, and not just sex hormones. Hormone is any chemical signal that travels from basically from a gland to a target site. So any chemical signal that travels in the blood is considered a hormone. The blood is going to maintain body temperature and distribute heat. And I think I talk about this later on, but just to give you a little preview of that. I think I actually talked about this last week. Yes, when we talked about skin. When you get hot, the blood vessels expand and you swell. And then when you get cold, the blood vessels are going to constrict. So it's going to play a part in maintaining body temperature. Maintaining pH. There's buffers in the blood that keeps it at a pH of 7.35, slightly basic. And to maintain fluid volume and to prevent blood loss. So if you have an injury, your blood's going to clot so that you don't have blood loss. And you have white blood cells and antibodies that are going to also circulate in the blood. Okay, blood components. This is a really cool picture. I love this picture. It's so like action-packed. So your blood is going to be composed of plasma, white blood cells and platelets, and then red blood cells. So if you took a test tube with blood in it and you put it into a centrifuge and you spun it, this is how it would separate out. And that's actually what they do. If you donate plasma, that's how they do it. They're going to separate out the plasma from the red blood cells, and they'll keep your plasma, and then they'll put your red blood cells back. I don't know if any of you ever done that. You have to be 16 to donate blood or plasma, and I'm going to talk a little bit about blood donations in a second. Or in a few minutes. And so your plasma consists, it's 55% of your blood volume. It's mostly water, but there's also ions, proteins, gases, nutrients, waste, and hormones. And then you have the actual cells. So you have your red blood cells, which carry oxygen, your white blood cells, which play a part in immune function, and your platelets, which are going to play a part in clotting. And remember when we talked about stem cells, you have stem cells as an adult or as a young adult that can differentiate into red blood cells and that can also differentiate into bone. So you have stem cells in your bone marrow that are going to develop into blood cells. So you have plenty of those. All right, so that's plasma and cells. Next page. So here's how blood cells are formed. Remember I said stem cells? A stem cell is a cell that doesn't have a job yet. It's stored in the bone marrow, which obviously, as the name implies, it's in the bone and kind of that spongy liquid part of the bone. And it's going to undergo a process called hematopoiesis, is where blood cells are going to form from stem cells. So it occurs in the red bone marrow, and the skull, pelvis, ribs, sternum, humerus, and femur. And so you know where the skull, the pelvis, the ribs, the sternum is in the middle of the chest. Humerus and femur, that's going to be your leg bones. And so these are just the ways that stem cells in the bone marrow can differentiate. So it can differentiate into a myeloid stem cell or a lymphoid stem cell, and then it's going to further differentiate into, so an erythrocyte is a red blood cell. Neutrophils, basophils, eosinophils, and monocytes, those are all macrophage white blood cells. And so we'd have to really get into the immune system and how the immune system functions, but just as kind of a quick and dirty version, these types of white blood cells are non-specific in what they attack. So they'll attack anything that's foreign, all right? And so that's what these stem cells are going to differentiate into are these white blood cells that are like your big eaters, are going to attack and eat anything that's foreign. Platelets, which are involved in blood clotting, and then erythrocytes, your red blood cells, which are involved in carrying oxygen. Your lymphoid stem cells are going to differentiate into B cells and T cells. Now that's still part of the immune system, and those are still considered part of your white blood cell count, but B cells and T cells are part of adaptive immunity in that they have a memory of a previous exposure, and they're either going to release, B cells are going to release antibodies, and then T cells are going to kill infected cells. So while these guys right here, these white blood cells right here, are going to actually eat pathogens, B cells and T cells are your white blood cells that don't eat pathogens. They're like more sophisticated. They're specific. B cells can release antibodies. T cells are going to kill host cells that have been infected. And I know that is like, that's kind of a lot of information about the immune system. I'm not going to ask you about that, but just in case you were wondering what the differences were here, I mean, that's a whole nother like chapter. And it is very action-packed. And like I said in a previous lecture, that's something we're not going to do in this class, but if you go on to take AP Biology, you'll get that. Okay, so let's talk about erythrocytes. So they're red. They're the red blood cells. They transport oxygen. They are shaped like biconcave discs. They have no nucleus because they don't really need to make any proteins. They have a hemoglobin. That's the only protein that they really need. Hemoglobin is an iron-containing protein that binds oxygen and then releases oxygens at different organs. Red blood cells have a lifespan of 100 to 120 days. And then remember, after that 120 days, I'd mentioned this in our Zoom call, red blood cells are then going to go to the liver. They're going to be basically like digested and chopped up. And then there is a pigment called urochrome. Sorry, it's urochrome. And that's the pigment that makes your urine an actual yellow color, which is kind of interesting because I'm sure we've all wondered that at some point, like, where does the yellow come from? It comes from your red blood cells and a pigment that gets released when they get digested. So some red blood cell disorders that a person could have is anemia. I know you guys have heard of that. So anemia is going to result in a decreased oxygen carrying ability. So normally that's going to be an issue with hemoglobin and a lack of iron. It could also be a low red blood cell count. But normally, to treat anemia, a person is going to take an iron supplement, really boost the iron content in hemoglobin, and hopefully be able to carry oxygen a lot better. So you can imagine if someone has a decreased oxygen carrying ability, decreased oxygen to organs is going to result in fatigue and getting tired very quickly and breathlessness. Sickle cell disease is an abnormally shaped hemoglobin. So while anemia is not enough hemoglobin, sickle cell disease is when a person has enough hemoglobin, which is the protein that carries oxygen on a red blood cell, but it's abnormally shaped. And that has to do with a genetic mutation so that there's a wrong amino acid in the protein. Therefore, the protein is not folded correctly. And then you have a sickle-shaped red blood cell. So like I said, it's oh, and good, we have a picture of it. So it's a genetic disorder. It is a homozygous recessive. So in order to have sickle cell disease, the person would have to get the mutation from mom and the mutation from dad. And we know that sickle cell disease is most prevalent in regions of Africa. And you may know why, but in case you don't, I'm going to tell you. In Africa, if someone has one copy of the mutation, So remember, to have the disease, you have to have 2 copies of the mutation. So if they have 1 copy of the mutation, that means they are a carrier of the mutated gene, also known as the mutated allele. So they have 1 normal, 1 mutated. They're gonna still have normal hemoglobin, enough normal hemoglobin to where they don't experience this disease. But now they have this advantage. And the advantage to carrying an allele is that they are going to be resistant to malaria. And so that's why sickle cell is still a thing, because if you can imagine a person who has a normal gene and then the sickle cell gene who's able to resist malaria is going to be able to survive. They're going to reproduce, and then there's a 1/4 chance that their offspring are going to actually have sickle cell disease. We'd have to go through a bunch of Punnett squares to do that, but just so you know, I just want you to know what sickle cell is. I don't need you to know the genetics behind it. Just what is it? And like I had said, this would be a better topic to discuss under a genetics unit. And so I kind of think I just want to leave this at this. Just if they're a carrier of the sickle cell gene, they're more resistant to malaria. But I just really want you to know, yeah, that these are two red blood cell disorders. And if there's a disorder of a red blood cell, a person is not going to get enough oxygen to their organs. And both of these are going to have common symptoms, breathlessness, fatigue. I had a student that had sickle cell disease. He couldn't stand up to do labs. He had to sit down to do labs. So the treatment for sickle cell disease is just, it's basically just going to be like support, support for fatigue and whatnot. And then the treatment for anemia is going to be iron. All right, leukocytes. Leukocytes that's another name for white blood cells. These are your white blood cells. And we know white blood cells are part of the immune system. And the immune system is going to defend the body against infection from pathogens like bacteria, viruses, fungus, and parasites. And then the immune system is also going to attack cancerous cells. And so I think I said this in my last lecture. You may not have caught it, but there's a cell called the natural killer cell. It's a cell of the immune system, and its job is to kill cancerous cells, which is pretty cool. So when an area of tissue becomes damaged, there are these chemicals, like these alert emergency chemicals that get released, and leukocytes are going to respond to those chemicals, and those chemicals are there to help them locate the area of tissue damage. And so here are all the types of leukocytes or white blood cells. Neutrophils are the white blood cell that's going to respond first. Eosinophils are the type of white blood cell that are going to, they're all going to help fight any type of infection. But when I think of eosinophils, I think more of the white blood cell that responds to parasites, like protozoa parasites. Basophils are going to be responsible for releasing inflammatory chemicals like histamine. Lymphocytes, those are your T and B cells, and I kind of gave a confusing explanation of those a little while ago. B and T cells are, they're going to form memory cells, so this might help you with that. When you get a vaccine, you're getting dead, attenuated pieces of virus in the vaccine, and you're going to have a little bit of an immune response to it. And that immune response, the goal of a vaccine is so you make memory B and T cells. So the next time you're exposed to that virus, your body can launch an attack on it and you won't even know. So let's say you get the flu vaccine, your body then starts making B and T cells. Then the next time you are exposed, or when you're exposed to the actual flu, those B and T cells are already there and they're stored, and they'll launch a response before you even know that you've been exposed. So hopefully that helps you with lymphocytes. The monocytes, on the other hand, are already there, and they're non-specific, and they're going to help launch an attack on basically anything that's foreign. And so you might ask, well, why aren't the monocytes enough? Why do we need lymphocytes? They're just not. Just like when, an army goes to war, you have your first line of defense, your second line, your third line. It's the same thing with the immune system. The monocytes are going to be like your first line of defense, and then the lymphocytes are going to be your second line of defense. Hopefully that made a little bit of sense. All right, so a disorder of the leukocytes would be leukemia. And we know that leukemia, it can be seen in all ages, but this is one of the cancers that we hear about most often in children. The bone marrow becomes cancerous, and cancer is caused by DNA mutations where the cell cycle is unregulated. And what happens is, so this is normal blood, and here are your the red blood cells. Obviously they're red. The purple things are going to be your lymphocytes, like your neutrophils, lymphocytes, monocytes. And then you see in this picture that they've proliferated out of control. And so you might be wondering, well, why is this bad? You have more white blood cells, so more of an opportunity to fight infections. The thing with the cancerous white blood cells are that they actually are not functional. So if there's a mutation that's causing them to proliferate out of control, then there are other mutations that are actually causing them to not even do their job right. So what you have are these cells dividing out of control, and then they're crowding out the red blood cells, and they're crowding out the white blood cells that could actually work. So what could this lead to? Well, it could lead to not enough red blood cells, so not enough oxygen to organs, so leukemia could lead to organ failure. And then the other thing that it could lead to, if it's crowding out functional white blood cells, is it could lead to infections. And so that's why that's bad. So the treatment would be chemotherapy. Chemotherapy is when drugs are taken and those drugs are going to stop the cell cycle, so stop cells from dividing, and radiation, and then stem cell transplant. And so stem cell transplant would probably be a pretty good treatment, since we know that it's easy to get blood cell stem cells from adults. So there would be a transplant, and what they would do is they would inject it into the bone marrow to hopefully get some healthy white blood cells. So probably a combination of the three of these is what's going to have to take place. All right, so let's talk about platelets. Other composition of the blood. So platelets are cell fragments that are like irregular shaped bodies, because if you think about it, the job of a platelet is just to cause blood to clot and to kind of plug up an injury. And so they don't really need to function like a regular cell. And I think you have that there. So it's needed for clotting blood. So they're here, right here, you see a red blood cell, and then you see the platelets around it. So they're just cell fragments, irregularly shaped bodies. And then here's the platelets right here. You can see they're kind of irregularly shaped. And what they're going to do is they're going to just plug up a wound so that red blood cells can't like leave, and a person can't bleed to death. And so I think that last, clotting equals stoppage of bleeding. Oh, sorry, hemostasis. So hemostasis is what you're going to fill into that blank. It kind of looks like homeostasis. So that's why I kind of got confused for a second. I made these notes, but I still, when I see them sometimes I'm like, wait, what did I mean by that? Okay, so hemostasis is the stoppage of bleeding, and it involves clotting. And so here are the steps of blood clotting. First step is that the blood vessel is going to spasm, causing vasoconstriction. Vasoconstriction is like a squeezing. And you can see how that would help if there's a tear. Then if it constricts, it can help minimize that tear a little bit, so it's not stretching so much. The platelet plug forms, so the injury to the lining of the vessel exposes collagen fibers, and platelets adhere, kind of like glue. They release a chemical that makes nearby platelets sticky, and more platelets come in. So remember, we said this was a certain type of feedback. So more gets you more. So this is an example of positive feedback. And then after enough platelets are there, you then have a protein called fibrin. Fibrin comes in and it forms a mesh, kind of like that mesh that you saw in that video where I showed you how skin grafting is done. That video makes me like feel so nauseous every time. But fibrin's going to come in, it's going to form a mesh that traps red blood cells, platelets, and it forms the clot. Normally blood clots are going to form within 3 to 6 minutes, unless a person has something like hemophilia. And I think I have that in this next slide. Oh, that is such a cool picture. So this is a picture of red blood cells, and you have platelets in the fibrin that comes in and forms a clot. It looks like it's like stuck in spider webs or something. So disorders, if you have a thrombus, so a thrombus is going to be a clot that's occurring unnecessarily. So a clot that don't know why it happened, but it's forming in a place that you don't want it to form. So this is where that positive feedback is like, no, please stop. So a clot's forming in an unbroken blood vessel. A coronary thrombosis is where there is a blood clot in the one of the blood vessels of the heart. And so that's going to cause a heart attack, because what it's going to do is it's going to cause the heart itself Now, I'm not talking about the blood that's going through the chambers of the heart and being pumped into the rest of the body. The heart itself needs its own blood supply. It needs its own oxygen supply so that the muscles can contract. And so where this thrombus is forming is in the blood vessels that actually nourish and provide oxygen to the heart itself. Hopefully that made sense. So I've actually seen someone have a heart attack. I've actually seen two people have a heart attack. Oh, one of them did not live. He had a heart attack in front of me, and it's kind of a tragic story. And students always get really sad when I tell the story, so I'm not going to tell the whole story. But this was a friend, a really good friend, her brother. He had a heart attack. He had Down syndrome. Okay, I'll just tell the story. He had Down syndrome and very, 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 very bad. And he got really, really angry about something, and he just kind of flipped out and had a heart attack, like right there, and he didn't make it. The other person who's had a heart attack is my husband. So when my husband was 37, which he's 39 now, so this is 2 years ago. Oh my gosh, it hasn't even been 2 years. He was loading the dishwasher, and he said, "I feel weird." He said, "My shoulder feels weird, my left arm's going numb, and I just don't feel right." So it's like this feeling of panic and dread that they get. And you always think when a person has a heart attack, they're going to grab their shoulder and they're going to be in pain and then they're going to fall over. It typically, most of the time, I don't think it happens like that. It's more of just a weird lightheadedness, a tingling sensation down the left arm, and just a feeling something's not right. And so my husband ended up refusing to go to urgent care. So I was like, fine. And I had these two little babies I was taking care of. I was like, fine, if you don't want to go to urgent care, I'm giving the babies a bath. And so I was in the middle of giving the babies a bath, and he walked up and he goes, "Okay, I'm driving myself." And I was like, "Wait, what?" And then I have these two babies, so I didn't know what to do, and I didn't know what was going on. I did not know he was having a heart attack. And so he went to an urgent care, then drove himself to the ER, and then it turns out he was having a heart attack. And he had to get 2 stents. And so a stent is where they're going to go in and they're actually going to clear out the clot, and they're going to put something into the blood vessel that opens it back up. So that's my personal heart attack story, which, the reason why, so you're probably like, what's up with her husband? He was 37, he had a heart attack. The reason why is because he has a genetic disorder. So we now found out where can't metabolize cholesterol appropriately. And so if cholesterol is filling up the blood vessels, causing inflammation, that's going to cause unnecessary clotting around it. So cholesterol can actually kick off this process right here, leading to a heart attack. An embolus is where a thrombus breaks away from the blood vessel and it floats freely. And this becomes a huge problem when it goes to the brain. And an embolus can also go to the lungs and cause a problem in the lungs. So when a blood clot forms in a blood vessel unnecessarily and a piece breaks off and it travels to the brain, that's what's going to cause a stroke. And it's called an embolus. Hemophilia is a hereditary bleeding disorder. This is a disorder that is linked on the X chromosome. And I don't know how much genetics you got in cell bio, if you got very much at all. It's one of those things that they kind of throw in at the very end. It is a very, 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 very important topic, very important unit for biology. And so we would talk about hemophilia and how it's X-linked and it's recessive. And how it can lead to bleeding disorders where the person doesn't clot enough. And so if there's an injury or a car accident or a surgery, this person is going to potentially bleed out and bleed to death. So those are your blood clotting disorders. Very, very interesting, I think. Even more interesting than blood clotting disorders are blood types and blood grouping. I love it. All right, so an antigen. So we got to kind of talk about antigens. Before I get into that, okay. So think about the blood cell, and remember I talked about carbohydrates hanging off of blood cells and them functioning as like ID tags. So all your red blood cells are going to have certain carbohydrates that hang off of them, and that's what gives you your blood type. So if I have a blood type with a certain carbohydrate hanging off of it, and you infuse me with someone else's blood type that has its own unique carbohydrate hanging off of it, my body will recognize that as foreign. And so my body will recognize that blood group that's not compatible as an antigen. And an antigen is going to be any foreign substance that the immune system recognizes as non-self. And so all those white blood cells that I talked about a little while ago, they're going to attack the antigen. So that's why blood transfusions, you got to have something that's compatible. Antibodies are Y-shaped proteins that are going to attach to antigens. So that's part of the attack that can occur on a blood that is not part of your blood group that gets transfused into you. This is what happens when the immune system attacks that blood. It's called agglutination, and it's clumping. So the blood that is not compatible, that has its own carbohydrate that your body recognizes as foreign, is an antigen. Antibodies that are released by white blood cells attack that foreign blood group, and it causes agglutination to occur. So hopefully the flow of this information now makes sense. So your red blood cells, this should say it has surface proteins, but it should also say it has surface carbohydrates. So the A and B blood type are going to be carbohydrates. So if you can write that in, it's not just proteins, they're carbohydrates that are hanging off the red blood cell. The A antigen is going to be a certain carbohydrate and associated proteins. The B is going to be a carbohydrate and its associated proteins, and Rh factor is just a certain protein. So hopefully that makes sense. So there's a carbohydrate that's an A carbohydrate, a B carbohydrate, and then you have an Rh antigen that's a certain type of protein. Okay, so let's look at blood types. All right. So remember, the antigen is the carbohydrate that's hanging off of the red blood cell that gives it its ID. And then the rhesus factor is a protein that is on the red blood cell. So here are all of your blood types. So let's just talk about this for a second. So here is your A carbohydrate. Here is your B carbohydrate. Here is your Rh factor. If you have both A, B, and the Rh factor on the red blood cell, you have everything. You have all the ID tags on there. So therefore, a person with AB positive blood can receive blood from anybody. Because if you think about it, there's nothing that it could recognize as foreign. If it gets A blood type, it's like, hey, I recognize that. If it gets B blood type, it's like, oh yeah, I recognize that. And if it gets anything that has Rh positive, it's going to recognize that. So let's look down over here. O negative is the exact opposite. It literally has nothing on the outside of the cell to give it an ID. So it's like a blank slate almost. So you can imagine if there's nothing hanging off of the O negative red blood cell, anyone can get O negative blood. But then think about it, if you're the person that has this blood type right here, you cannot get blood from anybody except other O negatives. So hopefully that kind of made sense. I have a game for you to play. If you took my class last year, you already played the game, but it's still fun to play it. So you get to play it again. Izzy and Michelle, I'm actually talking to you. You get to play it again. And then if you're taking my class next year, you'll get to play it again then too. It's fun. I like it. So you are actually have patients, 3 patients that come in, and you have to blood type them to find out what their blood type is, and then you have to pick what they're compatible with. So let's kind of go through a couple of these. So AB positive is considered the universal receiver because it does not see any of these other blood types as being foreign. If you look at A positive, what could A positive possibly receive? We could receive something that has the A carbohydrate, this blue one, so it could get A negative. It couldn't get this one. 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 It couldn't get any of the ones that have the B antigen on it. So A positive can receive from O positive. Doesn't see anything foreign on that one. O negative, A negative, and other A positives. So hopefully that made sense. So B positive, what what could a B positive person, who could they get it from? They can't get blood from anyone that has an A antigen. So any of these ones that have blue on it, a B positive person couldn't get blood from that person. They could get blood from O negative, O positive, B negative, and other B positives. Let's go down here to one of the negative blood types. Let's talk about B negative blood type. So if they're B negative, they cannot get blood from anyone that has an A antigen, so that eliminates all 4 of these over here. Could a B negative person get blood from a B positive person? So I want you to ask yourself that. Could a B negative person get blood from a B positive person? They're both B, right? The answer is no, they cannot. Because if you look at this right here, if you infuse them with this B positive, what is their body going to see as foreign is this little black thing, the little black thing. So if they're Rh negative, they cannot get blood from someone that's Rh positive because that rhesus factor protein is going to be seen as foreign. Same thing with O positive. People are like, oh, I'm O positive, I'm the universal donor. No, you're not. You're not. You're a great donor. You really are, but you can only donate to people that are positive for the Rh factor. A B negative person could not get blood from an O positive person because that rhesus factor would be seen as foreign. So hopefully that made sense. I think the game will help you out with that a little bit. And if you're totally confused, actually I don't have this on my next slide, but on your next page you see blood type compatibility table. That's your little crutch. I really encourage you guys to try to do the blood typing game without the compatibility chart, because then that would really show your understanding. But if you need it as a crutch, go ahead, or play the game using the compatibility chart and then try playing it again without it to see if you really understand blood typing, and if you really understand how these antigens on the outside of the blood cell gives it its ID, and how a person's body can reject a blood type that is seen as foreign. All right, so the next page gives you that compatibility chart, and then it says, can you save the 3 patients using Chrome, allow Flash Player? Now, if you're having trouble, everyone can do it. I always have students in class, they're like, my computer won't let me do it. And someone turns to them, they're like, allow Flash. And then everyone can do it. So I know it'll work for everybody, so please, please, please do it. It's fun. And then you click on that link, and then you perform the blood type test on the patients, and then give them their blood transfusion. And then the other thing that you're doing, I think this page really honestly is out of order. This should be somewhere else in this This should be somewhere else in this handout. That's kind of annoying, but that's my fault, and I got these all printed. Actually, no, it's just duplicated, so it shows it later. So then you're going to also perform a blood type test on yourself and or one family member. I only gave you 2 cards. And I gave you 3 lancets just in case you mess up with the first one. So you don't have to do this exercise if you don't want to. There are some people that their vasovagal nerve gets stimulated when they see blood, and they cannot consciously choose to not pass out. It actually stimulates this nerve and their blood pressure drops. So if you are a person that's scared of blood, don't do it. All right, and this slide right here, you may want to take a picture of this. This tells you what your results would mean. Again, this is telling you what you do. Your anti-A is the blue liquid, anti-B is the yellow liquid, and then the RH is the clear one. And your card tells you where to put each one. So you use the lancet, you prick your finger, squeeze the blood out. You're gonna have to like milk your finger. Make sure it's not cold, because if your blood vessels are constricted, you're gonna have a really tough time getting blood out. So warm your hands up. Warm them up under like hot water or something, and then it'll flow out really easily. And then you're going to put a big glob of blood on each test site. You'll see that on the card. And then you just put 1 drop of the test chemical, blue, yellow, clear, and then you take the corresponding toothpick. Do not use the same toothpick for all 3 tests or you're contaminating. So use a blue toothpick to mix up the blood with the blue. Use a yellow toothpick to mix up the yellow with the blood. And then I want to say I actually gave you a red toothpick for the RH factor one, and then you mix it up. Turn the toothpick on its side to mix up. I see students doing it with the tip, and then they fling blood everywhere, which At home, that's probably not a big deal. In the classroom, it's kind of unfortunate when that happens. So just I will show you on the Zoom call. And then this just shows you the prevalence of each blood type. So the most uncommon blood type is going to be your AB. Students are always so disappointed when I do their blood typing and they're not AB. Everyone wants to be AB, and I'm so curious to find out if any of you are, but most of you are probably going to be A. I'm A negative, so I'm kind of rare. I'm kind of special. B negatives, but these two right here, O positive and A positive, I would say out of all the students I test, I get A positive the most. So that'll be interesting to kind of find out where you are on that. All right, so the vascular system. I need to go through blood vessels pretty quickly here. So the vascular system, blood's going to circulate in the closed transport systems. You have arteries, which take blood away from the heart, oxygenated blood away from the heart. Arterioles, capillary beds, venules, and veins. So the arteries are going to take blood away from the heart. Arteries then turn into arterioles, which then join into capillary beds, which then turn into venules, which turn into veins. And so you have your large, medium, tiny, medium, large. And I have a picture here. I didn't put a picture in your notes, but this can kind of show you how your arteries are going to branch into arterioles, then into capillaries, then into venules, then goes into veins. And this is where diffusion of oxygen and nutrients and stuff is going to occur into organs. And obviously it changes from red to blue because this is right where hemoglobin is dropping off oxygen to organs and muscles and all the other parts of the body. So like I said, blood is taken away from the heart by arteries. They're thicker. They can withstand high pressure. And the reason why arteries have to withstand high pressure is because, think about it, the heart's right here and it is pushing really hard. And so arteries are going to be able to withstand high pressure even more and have thicker walls. Capillaries are teeny, teeny, tiny. The walls are like 1 cell thick, and that's so the gases can be exchanged between the blood and the tissues. Just like I said, this is where hemoglobin is dropping off oxygen. And then veins are going to be where blood is brought back to the heart. The walls are a little bit thinner, and the pressure is a little bit lower, and there is a larger lumen, meaning that they actually can hold a larger blood volume. And that makes sense because the pressure is a little bit lower. And the reason the pressure is lower is because it is bringing blood back to the heart, and it's not actually undergoing the pressure of the heart, like an immediate pressure of the heart contracting, pushing blood out. And there's valves in veins to prevent blood backflow, because if you think about it, blood's pushed from the heart down to the bottom of your feet, and then it has to come back up. So that makes sense that if it's coming back up to the heart and then to the lungs, that it has to have valves to prevent backflow. And then skeletal muscles are going to enhance venous return, so there has to be some help to get the blood back up. And so the skeletal muscles are actually going to help with that, which is pretty cool. Let's see, I don't know if you've ever seen like an older person that seems to have like bulging veins in their legs. I know that really sucks and that's gross. But as you get older, these valves that prevent backflow, they actually start to function, to not function as well. And so that's why people get like backflow and like swollen blood vessels in their legs. It really sucks. All right. I meant to change this picture and I meant to put a different picture. I don't know why I put this on here, and I looked at it and I was like, I just don't know if that's necessary because we're not even talking about the reproductive system. So I'm sorry I kept it there. We are all mature people and we can handle this, but it is distracting. So anyways, these are the places on the body where you can actually feel the pulse, which is the expansion and recoiling of the artery with each beat of the left ventricle. And we're going to talk about that tomorrow when we talk about the heart contraction. So these are all the places on the body where you can actually feel a pulse. And so on the picture I said, can you feel the pulse? So it would be cool if you would go through each one of these pulse spots to see, can you actually feel your pulse through them? And then your pressure points are going to be your carotid artery and your radial artery. Sorry, yeah, my screen went off. And those are going to be the two places, the carotid artery and the radial artery are going to be the two places where you're going to feel the pulse the best. And normal resting beats per minute are going to be 70 to 76. This does depend on your height, your size. So someone who's probably under 5 feet, yours is going to be upwards in the 80s and 90s. And then it also depends on your athleticism. If you're a runner, your pulse will probably be more down in the 50s. And so it just depends on situation. Our other vital sign that we're going to deal with tomorrow is blood pressure. So blood pressure is going to be the pressure of blood on the inner walls of the blood vessels. You have two numbers. You have one, it's like a fraction, but you're not actually dividing anything, but it looks like a fraction. You have one number on top and one number on bottom. The number on top is your systolic blood pressure, and that's the pressure that is exerted on the blood vessels at the peak of ventricular contraction. And diastolic blood pressure is the pressure exerted on vessels when the ventricles relax. So obviously systolic pressure is going to be higher than diastolic, but you have to watch both of them because you don't want either of them to be too high, because that can be an indication that there is cardiovascular disease. And so when you're at the doctor's office and they're measuring your blood pressure, they're going to look for the two numbers, and the numbers that are usually accepted as normal would be 120 over 80. I put 70 here because I think sometimes the people go back and forth on which number it should be. 120 over 80, 120 over 70. Obviously 120 over 70 is going to be a little bit better, but you definitely want it to be less than 120 over 80. And so when you go into the doctor's office or you go to CVS and you sit down in a chair, you're waiting for a prescription, you can use this manual or this this automatic machine right here. The automatic machine is going to be really easy. You just put the cuff on and then you push start, and that'll give you the two numbers. I'm going to have you guys use the manual cuff with the stethoscope because I think it'll help you understand a little bit more about blood pressure. And then also it's definitely a good skill to have if you're going into healthcare, because if your numbers are out of whack on here, they need to be verified with the manual stethoscope with the cuff. And this is just showing a picture of the setup. There's the cuff. This is the bulb that you're going to squeeze to inflate the cuff. This is the sphygmomanometer, I always mess up on that, where you're going to actually read the pressure, and then you're going to have the stethoscope in your ears listening for the heartbeat. So you wrap the cuff around your arm, you place the stethoscope on the brachial artery, like this guy's doing right here, or this lady's doing right here. It could be a man or a woman. You inflate the cuff to 180. You guys will probably inflate yours to 150. Unless you know you have hypertension or you have a history of hypertension, you're going to probably inflate yours to 150. You slowly release the air. If you can see where my cursor is right now, you see this little metal thing? The way you release the air is that you slowly turn it. And then you slowly turn it, air starts coming out. You're reading this while listening. When you hear the sound begin, you make note of where the dial is. Then you keep listening. When you hear the sound disappear, you make note of where the dial is. And so the first sound that you hear is your systolic pressure that you're reading on this sphygmomanometer. And then when the sound disappears, you read the number on here, that's the diastolic. So that's how you're going to do that. And you guys have a couple of videos. And so when there's homeostatic imbalance in blood pressure, a person could have hypertension, which means they have high blood pressure, which means they're going to have multiple reading over several days of 140 over 90. Circulatory shock is when a person has hypotension, meaning their blood pressure is too low, and this can be a result of blood loss. And then atherosclerosis is when the artery walls are going to thicken due to fatty deposits or plaques. And what that's going to do is that's going to lead to hypertension. So hypertension could be a sign of atherosclerosis. And then remember, atherosclerosis can also cause problems with blood clotting. And we all saw with the heart attack and the embolism how the blood clotting can become a problem. So definitely hypertension can be an indication of atherosclerosis. So that's why I definitely want, when we get older, we definitely want to have our blood pressure monitored and all of that stuff before, so it can be fixed before something bad happens. All right, so your assignments today are going to be the blood typing, the blood typing, actual blood typing, the blood typing game, and then to get started putting your sphygmomanometer together and your stethoscope together, and to start watching those videos and getting ready to do blood pressure measurements tomorrow. And I made it under 45 minutes. This was a long one, but I think you got some useful information, and I will talk to you guys tomorrow.